We're going to talk about tools for seeing biology. This will set us up to think about biodiversity and other things, but this is really just about tools for seeing biology. So let's get into it. Earlier in class, we talked about living matter and what makes it unique from a bioengineer's perspective. I want to focus on the last thing that we introduced, and it has to do with tools and tools of our time, 21st century tools. And when we think about seeing is a tool related to measurement or observing. So we're going to highlight that and really think about what we're doing in terms of observing or seeing or measuring biology. Earlier, we saw an example, incredible video from Grant Jensen's laboratory at Caltech in Pasadena, California, using an electron microscope to create a representation to see what's going on in a bacterial cell. This is one of my favorite examples because this bacteria, if you remember, actually grows magnets or magnetic crystals in an array that helps the bacteria react to magnetic fields. It's a magnetotactic bacteria. And so somehow we go from this microscopic organism to a fancy computer graphic that lets us see what's happening in the biology. And of course, if you're thinking about being an engineer, imagine being an engineer with a system where you can't see anything. You could do it, but it'd be tricky. It'd be much easier to be an engineer if you can see what's going on. So that's why we care about seeing and observing and tools that let us see. Now, so far, it might not make any sense at all how these sorts of images are generated, so we're going to dig into that. The technique has a fancy name, electrocriotomography, which is basically cold temperature electron microscopy with a tilting sample, tomography. So cold temperature, that's cryo, electron microscopy, tomography, tilting sample. And if you go down to Pasadena, they're happy to welcome you at Caltech to their cryo-EM facility, cold temperature electron microscopy facility. And within that facility, according to their website, you see an instrument that looks like this. Looks complicated. Looks big. Looks expensive. You know, these instruments, these cryo-EM machines, fill up a room. They cost $1 million, maybe $10 million for one microscope. $10 million, $7 million. Whoa. How do they work? We're going to observe a sample using an electron microscope. We'll explain that in a little bit. And the sample is going to have to be in a vacuum. So the electrons in the electron microscope get right to the sample. Now, if I put something in a vacuum and it's alive, like how would you do if you were in outer space? If you were like in a spaceship, you're like, I'm out of here. And you didn't have a spacesuit on, you just like went out of the spaceship and you're in the vacuum. My guess is you already know that you wouldn't do well. But why is that? Well, if in outer space the pressure is low, there's a vacuum. Think about what happens to water when you put water in a vacuum. You lower the boiling temperature of water. The water will start to boil at lower and lower temperatures, the lower the vacuum. In space, of course, water's just going to boil off. If you're alive, living things have water inside them. So if an electron microscope, the sample's in a vacuum, that it's like, how are you going to do in outer space? You're gonna like, are you gonna explode? No, not really. You're gonna burst though. And so you don't want that to happen because you can't really see what the thing looks like if it blows up, if it bursts, right? The thing that's full of water in a vacuum, quote, explodes or bursts. To solve that problem, one approach is to freeze it, right? And so you could freeze the living sample quickly to fix the water so that the water can't, evaporate into the vacuum. It will eventually, but not in the same way. And so the way this works is to use something really cold, something cryogenic, hence cryo electron microscopy. Cryo is any temperature below negative 153 Celsius. Well, liquid nitrogen, that's convenient. If you cool down nitrogen so it's a liquid, it's negative 195 Celsius. Practically what you do oftentimes is you'll first put your sample very quickly you know, for example, if you grow an ice crystal slowly, if you let it form slowly, you'll get big crystals. You don't want that to happen because that'll disrupt the sample too. So if you're going to freeze your living sample, you've got to go like a plunge, flash freeze. And so the first thing you do oftentimes is you don't go right into liquid nitrogen, you go into liquid ethane, which isn't quite as cold, but can absorb more heat. And so it's a faster way of cooling something down to quickly freeze it until you do a quick like plunge or dunk into liquid ethane and then move it over to nitrogen, liquid nitrogen, and then put it in. Wow, it sounds complicated. But okay, so just give you a sense of like how the Jensen lab is creating these microscopic, amazing images of cells. Now the next thing is we're going to tilt the sample, right? So if you're looking at something, you're like, look at your hand right now. What you're seeing is, you know, the light reflecting back off your hand, and you've got two eyes. So let's cover one eye, right? And now we're seeing the light, and your hand is a pseudo two-dimensional object, right? Even though you know your hand's a three-dimensional object, right? And so if we wanted to create an image of a three-dimensional object, but we can only see it in two dimensions, well, one thing we could do is we could move where we are with relation to the object. And it's like, oh yeah, if I look at my hand from this perspective, I get a different image, right? But in this microscope, instead of moving the microscope around, we're just going to tilt the sample. That's tomography. And by tilting the sample, we'll be able to get images from different perspectives or slices. That's what's shown here. And if we collect all of those images and keep track of them, then we can use a computer or software to infer and reconstruct what the three-dimensional object looks like. That's called tomographic reconstruction. This is used in microscopy and some types of medical imaging, right? But gives you a sense of, so now we have this fancy word, cryotomography, cold temperature electron microscopy with a tilting sample. And we have a sense of what's going on. But let's dig in a little deeper just to make sure we have a good foundation for how seeing things relates to bioengineering. And to do this, we've got to remind ourselves, like, how do we see things in the first place? Like if I'm looking at an apple, how come I can see an apple on an apple tree outside? That's because there's sunlight, and the sun is emitting various forms of radiation, for example, visible light. And that light has different wavelengths. When it encounters an object, some of that light can be absorbed by the object, reflected by the object, refracted, or passed through the object. And if light's, for example, reflected by an object, it can go into my eyes or your eyes, right? And in your retina, you know, different wavelengths will be detected. So an apple, for example, will absorb visible light except for the red, and then that gets reflected, and oh, an apple's red, or it's a green apple, you know, it's reflecting the green and absorbing the rest. So radiation hits an object and can pass through or reflect, and so on. So that's how we're seeing things. This radiation comes in different wavelengths. So let's think about how wavelengths determine what we observe and why we might be interested in, as bioengineers, in an electron microscope in addition to a visible light microscope. So here's a really cool video that helps us understand how the wavelength of light is related to the resolution of what we can observe. Now, what's resolution? It's resolving things, right? So if I'm looking at two objects, do I see them as two separate objects that I can clearly, you know, separate from one another? Or if I can't tell them apart, then I can't resolve them, right? So like, how far apart do two things need to be in order for me to see them as being two separate things? So this is a really neat video to illustrate the point. I've got an object that is just a pattern of lines, and the lines are some distance apart. And right now, because we have a whole bunch of different wavelengths of visible light looking at this microscopic object, we can see the lines as being different from each other. But in this clever example, what's going to happen is we're going to use green light, right? So suddenly we're only looking at this object using green light, which has wavelength, and we can still see it, right? We go to red, it's a little bit harder to see, right? Red wavelength is a little bit longer than green, not by much. What is it, like 100 nanometers longer in the wavelength? And because of that, it's much harder to resolve or tell apart the lines here, right? And the further out we go into the red, like suddenly we can't, it's the same object. The object's not changed. The only thing that's changed is the wavelength of light we're looking to observe the object, and whether or not we can tell apart features within the object. And so as the wavelength has gotten longer, we're not able to resolve the fine details in the same way, all right? So there's this relationship between the radiation and the wavelength of the radiation and how it reacts with the object and how we observe it. And the longer the wavelength, the bigger things have to be, the farther apart things have to be for us to see them as a part. Conversely, the smaller the wavelength, the shorter the wavelength, in other words, the better resolution we're going to get. So the nice thing about wavelengths is there's plenty of wavelengths. And in bioengineering, the most relevant wavelengths right now, at least, are the visible wavelengths, which are shown here. And so if we use a light microscope, what we mean by that is visible light microscope, we'll be able to resolve objects that are, a couple hundred nanometers in resolution. And here you see like red is a little bit longer than green. And so that's why green in the example, cleverly set up example, was able to see the tiny distances between the lines. But we were talking earlier about an electron microscope. So different type of radiation and so a different wavelength. And what's so interesting is the wavelength on an electron microscope is going to be way down here. And if you come down to the bottom of the slide, you can see that these wavelengths, if the visible wavelengths are, somewhere between, say, a virus particle and a whole cell, that means the visible wavelengths on a light microscope are going to let us see microorganisms and some of the structures inside microorganisms. But now if we jump down here, several orders of magnitude shorter wavelengths to electron microscope wavelengths, come down here, it's like, oh, those wavelengths are between the size of a molecule and the nucleus of an atom. Thus, we should be able to resolve objects and structures that are much closer together. All right. So let's look at another video of an electron microscope. Again, we'll stick with the Jensen lab, amazing group, super inspiring researchers. And let them walk us through how they can help us see what's happening in a living cell. All right. The first thing we're going to look at is the sample frozen in one of these fancy microscopes and it being tilted and the images being captured. So you can see the tilting. There's a bacterial cell in the middle and it's being tilted back and forth. Isn't that incredible? Let's look at that again. You might notice all these dark dots all around. Those are typically gold dots, like physical particles of gold or something that the electron microscope, the electrons will really interact with. And so it's very easy to tell what's there. And these are registers or markers that the computer software can use in the tomographic reconstruction to help understand where things are in three-dimensional space and create a more accurate three-dimensional reconstruction of the cell. So those are purposely added to the sample when it's frozen to create a set of marks or indices that make it easier to do the 3D reconstruction. All right, so that's the tomography part. Now we're going to do computational reconstruction. So let's see what that looks like. instead of tilting, we're just representing it as an up-down series of slices. And so now it looks like this is what the microscope's seeing, but this is actually a computational reconstruction of all the tilts to give you a stack of slices from the raw data. Now that we've constructed this three-dimensional representation from the tilted slices, we're going to start to identify biological aspects of the cell. How about the membranes encompassing the cell? We've got an outer and inner membrane. We're going to identify those structures in purple and blue. Remember, this is an electron microscope that's got resolution between nuclei and molecules. And so, it looks like, if you just look at the image, it's like, oh, it looks kind of grainy, but we're going to zoom in and pick up on the detailed resolution of the microscope, right? So within this membrane, oh, we've got some peptidoglycan, right? We've got these structures of atoms that are part of the membrane system surrounding the cell. The nucleoid, as a bacteria, you're not going to have a nucleus, but you're going to have a region where the chromosome is condensed or localized. And so you can begin, that's the DNA in other words, and you can begin to say, well, what does that look like? Where's that? Right? And here they're highlighting that and using the computer to identify that. The flagella is something that's typically sticking out of the cell and pushes the cell through the environment. It pushes the cell by spinning around. To spin around, it has to have a motor. The motor is made of proteins. And here's what that looks like. And by the way, you have microscopic motors inside cells pushing cells around, right? Like that sounds like engineering. If you're going to be moving around, you need to have a sensor array to know where you want to go. You're going to be sensing chemicals. And so here are the things on the surface of the cell that are interacting with molecules in the environment. And depending on what's interacting with the receptors on the cell surface, you're going to control your motor moving the flagella, pushing you around or tumbling. Maybe you have to have things coming in and out of the cell, pores and other stuff, right? So it's like pretty cool, right? It's just absolutely amazing. These microscopic objects made of molecules that turn out to be cells have all these different parts, and using electron beams in a fancy electron microscope, we can take a look at what these things look like. And once you get a sense of what they look like, it feels a lot easier to be a bioengineer. It's like, do we want this thing over there? What if we changed it? What would that look like? Right? Ribosomes. Oh yeah, those are the things that make proteins. Where are they in the cell? Oh, they're around, where the DNA is, and we can count how many there are, and so on and so forth. So you get the idea. So I just want to take a deep dive into something that sounds really complicated and totally esoteric, which is valid, but hopefully you have a sense of, well, this is what 21st century bioengineering is enabled by, right? Okay, good. However, what are some of the limitations of this microscope? First off, it's really expensive. I don't have one of these. People on my team, they get to use it, but we have to go like beg or borrow for access. And it's like really grateful we can use it. It's like, whoa, who can afford a $10 million microscope? Hardly nobody. So it'd be nice if this cost like $10. Would that be possible? Another thing is, well, this is frozen. And so it's not really alive. I'm like seeing everything in one fixed time. And so I'm not really sure how it works. I can see it like, but what's really behaving? It'd be like having a photograph of people playing soccer. And you're like, how does soccer work? But it's just like one image and you don't get to see the players running around, right? It'd be like, do you really understand soccer if you could just see one image on the field? I'd rather have something that's not frozen, but I have to freeze it. Otherwise the living cell is going to get blown up when it's in a vacuum. So how do I get a non-destructive image, right? And the other thing I didn't talk about is this electron beam that's coming in, that's a form of energy, and it's basically going to be cooking or damaging the sample. And so I got to be careful about how much energy I'm putting into the sample. Otherwise, I'll destroy it as I'm trying to see it. And so I'd like to have an affordable microscope like this. I'd like it to be non-destructive, doesn't require freezing, doesn't use too much energy. And I want to see this living thing. And also, if you remember from our diffusion conversations, these molecules, when they're moving, we know how fast they're moving. The time between molecular collisions is about a nanosecond, which means I need a non-destructive microscope with this level of resolution that's observing the entire cell with gigahertz frequencies, like a billion times a second. So give me that. Right now, if you give me that, like now we're talking, now we're bioengineering. But of course, that type of microscope doesn't exist yet. Parts of it may be physically impossible to make. You should talk to your physics friends, right? Hey, could you give me, cryo-electron tomographic microscope that's got gigahertz sampling rates and is non-destructive? And they're just going to go, what? But like, keep asking them. All right. What do I mean by this? I want to zoom out for a second. One of the unique and incredible things about 21st century bioengineering is it's an emerging engineering discipline, which means, what do we mean by emerging? Like, we have an opportunity to advance the discipline, not just by doing useful things for patients and the planet, but also by improving our tools, making better tools for bioengineering. Ever better tools and approaches are essential to advancing bioengineering. So this type of work in and of itself, tool development, tool improvement, is part of bioengineering. What am I talking about? Well, on the middle of this slide, you see a simple schematic: design, build, test. No matter what type of engineer you might be, you could be a mechanical engineer making a car, or a civil engineer making a bridge, or a chemical engineer making a refinery, or an electrical engineer making a computer, you name it. No matter what type of engineer you are, once you've decided you know what you want to make, you got to design the system, then you got to build the system, and then you got to test the system. Is it working, right? When we talk about making better tools, we're talking about improving the ability to design, build, test. And any improvement you make in this cycle is just going to have a sustained impact on the engineering workflows, right? Like, a good engineer is able to make a useful system that people can rely on. A great engineer makes engineering easier. And one of the ways you make engineering easier for everybody is you improve the tools. All right. So, we've already talked about in this class a type of tool called DNA synthesis. Well, that's an improvement because it could allow you to accelerate the step from design to build, right? We've talked about the idea of testing and measurement and standards. We've talked about the idea of abstraction for managing complexity. These are, over the last 20 years, types of categories of improvements, but more simply and abstractly, like better tools for measurement, better tools for seeing, better microscopes, better bioengineering, better tools for modeling, representing cells, representing molecules, better bioengineering, better tools for making, better DNA synthesis, better tissue printing, which we'll get to, better bioengineering. So I just want to pause in this zoom out and talk about this particular aspect of 21st century bioengineering, which is 21st century tools, and the culture that celebrates and supports and rewards tool developers. You're probably now thinking I sound like a broken record on this topic. The reason is it's very difficult in bioengineering to work on improving tools. It's not that it's technically difficult to make better tools, it's that it's culturally difficult to focus attention on making better tools, because I bet every one of you is first interested in bioengineering, not from a tool perspective, but because you care about somebody who's sick, or you care about an endangered species, or climate change, or something wrong with the world, and you want to solve a problem that's an immediate problem, and it's totally valid and important. I love that connection. Never give that up. But bioengineering is so powerful for solving these problems. It means that bioengineers tend to be exclusively focused on solving the problems right now. And it's very difficult to get attention and support. So you know what? Instead of solving this disease, I'd like to make it easier for all disease to be solved. And to do that, I need to work on tools. Do you see what I mean? So foundational science and foundational tool development is very tricky to protect and celebrate as an activity. That's why I'm spending a little bit more time than usual on this one slide. Hopefully you get it. Now, to wrap up this, I want to transition back to light microscopy and talk a little bit about how tools are not the same, right? And I'm using funny language, like tools are not tools. What I mean by that is it depends on the form that the tool takes. The particular form that a tool takes impacts who can use the tool where for what purpose. Here's an example in the 1960s, 1970s of a computer, an IBM mainframe computer, an industrial computer. The people who could use this computer were people who could afford computers in big rooms, right? And certain people could do that. Shortly thereafter, around the same time, a different type of computer showed up. The personal computer, just across the creek for where I'm recording this lecture in Menlo Park. It's still a computer, but it's not the same thing. A computer is not a computer. It depends on the form of the computer. What's interesting here is that the personal computer as a revolution is not a revolution in the physics of transistors, right? It's a revolution because of the word personal. It changed who could compute where for what purpose. Tools are not tools. A computer is not a computer. A microscope is not a microscope. A DNA sequencer is not a DNA sequencer, and so on. It depends on who can do it where for what purpose. What do I mean by that? A microscope is not a microscope. You know, what if a microscope, instead of costing $1 million, costs $1? Instead of being this fancy instrument in a room, what if it was paper? What if you could step on it? What if you could use it as a bookmark? What if everybody had a microscope? A personal microscope. Well, guess what? Here it is, the fold scope. The fold scope is an origami microscope. And each of you will get one, and you'll have to build it. And then you get to use it. Now, this microscope is an optical microscope. And so visible light will be the source of radiation that you use to see things. And so that means the lowest wavelength is going to be about 400 nanometers, right? So you'll be able to see things on the order of a micron, a little bit less than a micron, and a little bit bigger than that. This is a tool within the context of the PSET will encourage you to use to help you understand yourself. We're going to send you out into the world to look for biological things. And by doing that, you'll have some choices to make about what types of things you're looking for and why. It gives you experience with thinking about tools and improving tools. By going out and looking at biology outside of a laboratory, we're going to encourage you to think about nature and natural biodiversity and all the life that pre-exists us on Earth. As bioengineers, it's incredibly important to have empathy for the planet and the life that we inherit, so to speak. It's not ours, but it's arrived before us. What do I mean by that? You know, in computer science, all of computer science has been invented in the last century or so. The software that's on GitHub, all the code, it's like a year to a decade to a couple decades old. But I'm talking about biocode. If I'm talking about DNA code found on Earth, oh, you know, that's like 1 million years old, 1 billion years old. Right? So it's like back to nature, like, whoa. One of the amazing things about bioengineering is we're encountering this evolved code base, right? And then, of course, hashtag bioquality. What if everybody had a microscope? The fold scope is an invention of Stanford bioengineer Manu Prakash and his team. And it's so interesting in and of itself. I'd encourage you to take a look at Manu's website and see what's going on there and why they're doing it. Okay, so to wrap up, here are some questions to think about. What is resolution with respect to seeing? What determines the resolution limit of what you can observe? How could microscopes be made better? I gave you my perspective with respect to cryo-electron tomography. What do you think? I love this question. I still wrestle with this question all the time. At the molecular to cellular scale, is biology complicated only because we can't see it easily? Remember that idea of like a cell made as big as a building? Like if we could just see what's going on and observe everything, like suddenly it would just make sense. And we'd be bioengineering, like, ta-da, it's all working really well. Or is biology at this scale, molecules making cells, is it somehow intrinsically complicated? Even if we could see it with perfect resolution, with perfect sampling rates, like even if you gave me a camcorder, video recorder that let me see how molecules were making cells with like perfect resolution, perfect, would it still be complicated? I'm not so sure, honestly. I think it might actually not be that complicated. But what do you think? Thus, why is developing better tools important for bioengineering? And also, why is it hard? Why is it hard to organize resources to make better tools? If you go to your local, here's an idea. Write the person in your district for the US House of Representatives and say, I think it's important for making better tools for bioengineering, and here's why. And see what they write back. Why is it hard to make that argument? Last question. Wherever you are, look at different forms of technology. Maybe it's a screwdriver, maybe it's a computer, maybe it's a bicycle, maybe it's a car, maybe it's a food processor, a microwave, or an oven, or a stove, or a refrigerator. Like, what are some examples of different forms of technology? Challenge yourself to see, a refrigerator is not a refrigerator. It depends on the refrigerator. A bicycle is not a bicycle. It depends on the bicycle, right? Like, start to see technology not only as a label, but from the perspective of who can use it where for what purpose. And if you can see that extra dimensionality of technology, you'll be so equipped to understand how different forms of bioengineering tools will impact who gets to be a bioengineer. Thanks so much. Can't wait until next time.